So you've seen enough of me. Let's get started. We begin today discussing the placenta and the membranes. So we will step right into the lecture scheduled on the syllabus for today so that we progress and proceed on schedule as much as possible. So we've talked a little bit about the development of the interface between the developing human zygote, embryo, and fetus with the maternal side. And now we'll look at the details of that before we move on to the study of organogenesis and the organ system development. Everything still so far has been basically survey, and now we proceed to greater detail in the evaluation of these structures and their development. So the chorion, we recall, was extraembryonic somatic mesoderm plus 2 layers of the trophoblast, so the cytotrophoblast and the syncytiotrophoblast. The amnion is another fetal membrane, excuse me, and we followed its development from very early on from the trophoblast layer of the blastocyst, and eventually the two-layered disc, embryonic disc, germ disc, excuse me, began to become suspended in this 2-chamber space, the dorsal one being the amniotic cavity, of course the ventral one, the extracoelomic cavity, that is the primary yolk sac. So the amnion is a thin membrane enclosing the amniotic or the dorsal cavity, and after the embryonic folding, this sac surrounds the developing embryo just about 360 degrees. So we talked about folding and everything coming in to collapse basically upon the umbilicus, or the umbilical cord. So it is the junction on the ventral surface that occurs. So it starts in the dorsal and like a cape comes around and attaches on the ventral aspect of the developing embryo. The amniotic fluid is derived from the maternal secretion and the fetal fluid diffusion into this space. So it is a fluid-filled sac. And of course, this is the one that when it ruptures, the person says, oh, my water just broke. So it's the amniotic fluid that is the first presentation, basically, of that stage of delivery. So the amnion continuing then, the fluid moves in both directions, maternal and fetal. The fluid is primarily water, and the buoyancy of the fetus in the fluid is essential for the normal growth. So the fetus is suspended, the embryo is suspended up until the 8th week when it becomes a fetus. So there isn't any compression and distortion of development. So it is very protective in that sense as well. It is possible to have too much or too little, or I should say too little and too much, since they're presented in that sequence, of the amniotic fluid. So oligohydramnios is a low amniotic fluid volume from a low placental blood flow or rupture of the sac with leakage, or even possibly renal agenesis in the A meaning not, in genesis development, so the urinary system, kidneys especially, not even developing, so there is a urinary obstruction. There is this complete fluid exchange throughout all of these chambers. Once the fluid starts passing through the urinary system, of course, the only place for it to go is into the amniotic fluid. So polyhydramnios is the opposite, so a high volume of amniotic fluid. Typically, this is due to an esophageal atresia. So oligohydramnios is more going into the fetus, the late embryo fetus, than comes out through the urinary system. Polyhydramnios is some coming out, but not a sufficient amount going into. So this amniotic fluid cycles through the embryo. late embryo and fetus. So this polyhydramnios can be caused by a few additional things, such as gut obstruction, renal agenesis, the placental insufficiency, and growth retardation. Obviously, that was in a question format, not the other. I tricked myself on that one. So the gut obstruction really is the one that is included. So renal agenesis, placental insufficiency, and growth retardation are not causes of polyhydramnios. I apologize for that confusion. The yolk sac is the next membrane that we can discuss, and we've seen it develop. There was the primary yolk sac, the extraembryonic coelom, and the secondary or actual yolk sac developed within that area. So it is a significant structure until about the 20th week. It's necessary for nutrient transfer between the placenta that becomes established. It's incorporated into formation of parts of the gut and the respiratory tract. So we recall that it's lined with endodermal cells. And it is the site of the early differentiation of the primordial sex cells. So we will follow the origin of those primordial gametogenic cells and their migration to the posterior abdominal wall, where they will join with other cells, mesodermal cells, to produce the gonads. So this illustration is figure 722 from the textbook, and it shows how the amnion enlarges, fills the chorionic sac, and then envelops the umbilical cord. So let's take a look at that. We recall that the early three-cell layer, germ disc or embryonic cell disc, becomes suspended within the blastocele, the extraembryonic coelom, that is becomes referred to as the chorionic cavity once the chorion has developed. And we see the dorsal, the amniotic chamber, and the yolk sac chamber, or cavities, and the connecting stalk. So this extraembryonic mesoderm is present throughout this area here on the surface of the amnion and the amnion and the yolk sac. So the amniotic sac will change shape as we get the flat increase in length in every direction. If we think of it as a disc, we know it isn't perfectly circular, but in all dimensions, increases in length, and we get lateral folds, head fold, tail fold, and this pulls this cape down around to engulf basically the yolk sac, and then the entire amnion is the only structure ultimately that will be exposed into this chorionic cavity. So the connecting stalk is part of the extraembryonic mesoderm containing the allantois, the vitelline duct, as well as the umbilical and vitelline vessels. We had seen these, and will continue to follow their development and significance throughout the studies. Later, the amnion envelops the connecting stalk and the yolk sac stalk with their vessels, forming that primitive umbilical cord. And here we see this as it has occurred. So we see the yolk sac here in yellow. Again, endoderm. So the endoderm has now become the lining of the entire gut tube and the respiratory system, just as we had indicated before. So we see that amnion growing in size and occupying this chorionic space. And the body stalk here is becoming incorporated into this structure, and now is extending in length as the late embryo and fetus becomes suspended within the amniotic cavity and it continues to increase in length as the certainly fetus, now at this stage, is suspended fully within a greatly enlarged amniotic cavity. You see here a remnant of the yolk sac, so it does become dissociated and discontinuous from the gut tube. So what is the difference in the composition of the primitive and the matured umbilical cord? So the contents of the primitive cord. So I think it's obvious to see how these could be converted into exam questions. The connecting stalk that contains the allantois, the umbilical vessels consisting of 2 arteries and 2 veins, the yolk sac stalk accompanied by the vitelline vessels. So many of these structures will disappear as we get to the more mature umbilical cord. And so we then have only the 2 umbilical arteries, 1 umbilical vein, and Horton's jelly. So the objective, of course, is for you to do the subtraction procedure and determine what disappears or will no longer be found in the mature cord. So what are the names of the blood vessels of mature umbilical cord, and which of them transport oxygenated blood? So there are the 2 umbilical arteries and the single unpaired umbilical vein, and the vein transports the oxygenated blood. So this is where I threw myself off in that illustration a few lectures back. Went for the old blue and called it the vein, but of course there should be 2 blue structures, the 2 umbilical arteries carrying in the umbilical cord, carrying the deoxygenated blood in the waist to the placenta, and the 1 red umbilical vein bringing back the nutrient-rich, enriched, and oxygen-saturated blood. The allantois is a very interesting structure. It is a caudal diverticulum of the yolk sac, and it becomes a non-functioning remnant in humans, but it can cause some clinical problems if it persists to a great extent. It will contribute to the formation of the bladder, and it is also the site of those very early primordial gametogenic cells. What we will see as we follow this is that it will maintain a connection from the urinary bladder to the umbilicus, and in some females in the late stages of pregnancy, as there's obviously very much increased intra-abdominal pressure, there is the possibility for this to become patent again and droplets of urine to present at the umbilicus. I'm sure that would be quite shocking, even if you knew it was it's the persistent urachus that has become patent again. So now let's ask, what is the fetal contribution and maternal contribution to the placenta? So the fetal contribution is the chorionic sac and the villi that we've seen develop. We've followed their development, and we'll look at that in a little more detail as we proceed. It is visible from the maternal side as cotyledons, as they are referred to. The maternal contribution is the endometrium, referred to as the decidua basalis. Actually, there's a few different parts to it that we will describe. Decidua capsularis and parietalis are not involved in the endometrial contribution to the placenta. But there are other parts of it, the decidua capsularis and parietalis, that we'll define and describe as we proceed. So the endometrium contributes to the interface with the late embryo and fetus, obviously a zygote right through fetus, and becomes divided into the 3 layers. The decidua basalis, as we'll see in an illustration, is that associated with the cotyledons. And we now take a look at the other layers. We now recognize this as a coronal section, so going basically like taking a knife and cutting through the ear to ear plane right down through the center, and we're looking from the anterior view of the uterus. How might I be able to tell the anterior view? Because we said the most common site of implantation is on the upper posterior portion of the body of the uterine cavity. Otherwise, there wouldn't really be any way to tell whether it's anterior or posterior view. So we see the bulging conceptus from this view. It is now pushing back out through the endometrium. This is the 4-week embryo. So there is, it creates a bulge into the intrauterine cavity. So we now can look at the different layers of the endometrium, at least the two that we mentioned previously. They're not associated with the placenta. There is that covering the surface where the embryo is producing a bulge, so the endometrium will thin out in that area and forms a capsule over the developing embryo. We had introduced this term decidua for the cells making up one of the types of parenchymal cells making up the endometrium, and now we see this term persisting. So the decidua capsularis, if you cut that away, then you would see the chorionic villi on the chorionic sac on the opposing side. The decidua parietalis is the remainder of the endometrial lining other than the capsularis and what we see here now, the decidua basalis that interfaces with the cotyledons, forms these blood lakes, and we'll begin to focus on that a bit more as we proceed. A few other changes of the uterus are present and indicated in this illustration. We see that the intrauterine cavity will eventually become nearly completely occupied and, of course, ultimately fully occupied by the late embryo and fetus, and it is the amniotic cavity, amniotic sac, that is in contact with the decidua capsularis, of course, with the smooth chorion in between. So we now see that the amniotic sac in this panel D on the right completely occupies the chorionic space, chorionic cavity. So this obviously is part of the chorion interfacing with that portion of the endometrium, decidua capsularis. The decidua basalis, the decidua parietalis is all this other portion, and the decidua basalis is directly associated with the part of the chorion involved in the actual high volume gas and nutrient waste exchange. So we notice that the cervix is changed and the cervical canal is filled with a mucus plug. So this chamber has to be protected from the external environment, and that seems to be a function for the mucus plug. So we see that the decidua basalis really becomes extensively invaded in the fetal stage in panel D. So the fetal membranes then are the amnion, the chorion, decidua parietalis, and capsularis. We see these even at a later stage. The mucus plug persists in panel E in the later fetal stage, and the decidua basalis becomes much more extensive as the placenta grows in size. Relative to the size of the developing fetus, it seems to regress, and the villus chorion and the decidua basalis are still quite extensive, but the fetus has become greatly enlarged. And what we now are beginning to see is the interface of this hot layer or thinly stretched layer of the smooth chorion with the amnion inside of it, and the decidua parietalis. It takes position over the internal os of the cervix. And this area of the internal os of the cervix will begin to stretch. Everything begins to stretch, as we know. And the one barrier between the fetus and the cervix is this new structure after the decidua parietalis, capsularis, has degenerated, called the amniochorionic membrane. So it is made up of the amniotic membrane, the amnion, and the smooth chorion layer. So we recall that the cytotrophoblasts contributed to the formation of this portion of the chorion. The layers of the chorionic plate are the amniotic epithelium, the central mesoderm, the chorionic mesoderm, or the trophoblast. And stuck again. Okay, prompt us for follow-up on this. On a blank. Believe that well, I'm just not going to say what I believe. We'll just proceed. So prompt for replies on that. I do want to make a quick note. I know there's been some correspondence on the web mailbox on the WebCT, and for some reason, these are not showing up in my inbox. I believe that Marco might be reading them and replying them, and I don't know if that removes them from the inbox. But my other course online, I'm having a similar problem, so I'm going to check with WebCT on that. So I will check all the mailboxes, not just the inbox, from here on out. So let's proceed then to figure 7.6. Looking at a sagittal view. So this now is looking from the side, a lateral view of the gravid uterus, meaning the pregnant uterus. So the gravid, it means that there is conception has occurred and that there is a pregnancy ensuing. This is at 4 weeks. Seems to be a bit advanced, but anyway. We have the relationship of the fetal membranes to the uterus and to each other. Excuse me. And the amnion and the smooth chorion have been cut and reflected here to show these various layers. So we see the blue layer is the amnion, and the red layer immediately outside or peripheral to that is that decidua capsularis. And it should be immediately, the decidua, the smooth chorion and the decidua capsularis that they're not even showing in this illustration. And then the decidua parietalis will be the other portion of the endometrium. So these would be the two layers, the smooth chorion and the amnion on the embryonic side, and then there would have been the decidua capsularis overlying the chorion. To define the term decidua, so what is this really referring to, and what are its parts in relation to the implanted embryo? So this is obviously an immediate review of the information that we just looked at. So the decidua is that functional layer of the gravid endometrium. The basalis is between the chorion frondosum, so it has these fronds on it, and the myometrium. It is the primordium of the maternal portion of the placenta. The decidua capsularis is over the embryo, and the decidua parietalis is the remainder of the decidual endometrium. Now we can begin to consider the placental circulation. We had discussed briefly the development of the circulatory system in the embryo and toward the fetal stage. We now recall that we have the umbilical arteries that are very much involved and the chorionic arteries that are delivering blood to the chorionic villi, those fronds of the chorionic villi. So we will now proceed to the point of discussing this cell layer interface between maternal side and fetal side at the placenta. So the umbilical arteries will branch into smaller and smaller vessels until they encounter the tertiary chorionic villi, and this is how they provide this large surface area to perform their function. Sorry, I hit the wrong button. There we come. That's the top. Wow, trying to move my pip. Okay, the umbilical vein is obviously on the receiving end of things once these chorionic arteries break up into capillary beds. So this illustration, figure 7.7 from the text, allows us to follow this circulation. So two blues, we have fetal circulation in the umbilical arteries coming out, exiting and the embryo and fetus, and entering into the placenta within the chorionic plate. Hey, there's the chorionic plate. Looks like it's just the syncytio and cytotrophoblast layers that contribute to the chorionic plate. So we recall the syncytio and cytotrophoblast cells are involved in this area here, and also the chorionic plate, and also at the interface on these fronds of the chorionic villi. These obviously are tertiary, fully developed chorionic villi. So we follow the blue vessel, the artery in. There's a rich capillary bed on the tips of the fronds of these chorionic, tertiary chorionic villi. And then we can take it, and then obviously on the opposite end of the capillary bed is, in this case, the red venous drainage of those structures, bringing oxygenated blood back toward the fetus, late embryo and fetus. Now we switch to a quick look of the maternal circulation. So the endometrial arteries, we recall those spiral arteries coming into the endometrium. These now actually are open. So it's at this stage in pregnancy where we have the one instance of an open circulation in humans. Other examples of open circulatory systems like this, where the blood actually goes right into the interstitial fluid compartment and then somehow returns to the circulatory vessel, cardiovascular system, is in mollusks and those types of invertebrates. So the humans have a closed circulatory system except for this instance. These arteries are absolutely opened, and there's essentially a hemorrhage into this interstitial fluid compartment, and that is drained away by the endometrial veins. So this blood is squirted right into this area, and it bathes the tertiary chorionic villi. So this chorionic villus has to remain suspended and fully distended in this space where the blood lake is occurring. So there is typically one or more of these anchoring villi that anchor the tip of the chorionic villi, certain fronds, to the syncytio-trophoblastic layer at the interface with the decidua basalis of the endometrium. So this holds them in place so they don't just collapse and compromise the blood flow. So this illustration, figure 7.8, is showing us, again, let's stick with the blue, for the arterial input. Some branches as these umbilical arteries within the placenta or the chorion split. There are branches that go up into the tertiary villi and give further division and branching until a rich capillary bed, and then the venous drainage brings it back in the same direction into ultimately, of course, the single umbilical vein. Now we proceed to take a look at the illustrative, but still histology and cytology of these chorionic villi. So this arteriocapillary venous network can be seen if there is a cross-section through a chorionic villus. So panel B and C will take us through that discussion. So in an earlier stage of the development, we see the placental membrane presented here. We had seen this before where we talked about the initial development of the capillaries. Excuse me. So there is the syncytiotrophoblastic layer and the cytotrophic blastic layer. These are red, so venous side of the fetal circulatory system. These are blue, so the arterial side of the circulatory system, but this would be a capillary bed, deoxygenated blood coming in, oxygenated leaving. And then here in the lower panel, we can see that there will be the capillaries more closely adjacent to the cytotrophoblastic cell layer with larger vessels in the core that will be feeding into this rich capillary bed area. There obviously has to be a development. We recall that we had first the syncytio or syncytotrophoblastic layer, and the cytotrophoblastic layer grew into it. We likened it to having a clump of clay and pushing our finger into it. The extraembryonic mesoderm infiltrated that area like it grew into the inside of our finger, and then began to differentiate into the vasculature within there. This will grow and develop until the junction between the fetal blood and the maternal blood will become as thin as possible so that we have the most efficient exchange of gases and nutrients and waste. So the names of the vessels in the mature cord and which of them transport the oxygenated blood. So the umbilical vein is unpaired, and it is the one that is red in the illustrations because it's returning oxygen-rich blood back to the late embryo fetus. Now, what are the maternal components of this circulation? So that intervillus space contains the maternal blood in that blood lake. So it is actually, in a sense, this hemorrhage into the extracellular fluid compartment between the fetal side and the maternal side of the circulation. Their spiral arteries are the ones that are opening directly through their passage in the decidua basalis, and they'll discharge the oxygenated and nutrient-rich blood into this blood lake. And the spiral veins on the opposing side will drain off the fluid in this area. So the hydrostatic pressure is very important. That's why it all has to be enclosed and contained so that we do have a hydrostatic pressure. So whenever the arterial side or the heart beats, it has an output of blood, immediately blood goes into this blood lake area, and immediately, simultaneously, the flow is continuous into the venous side. So we don't get any clotting in this area, so it's obvious that this is a very important interface. So let's take a little bit of a closer look at this. On the upper left panel, we see the maternal side above with the gray shading. Uterine glands are still present. Here we see the uterine vessels, and they indeed open directly into this intervillous space and bathe the syncytio or syncytotrophoblast layer. Those two terms are synonymous. And then the cytotrophoblastic cell core is within this. So at this stage, we would have this type of an appearance with the early vessels being formed. Remember, this mesodermal layer is going to infiltrate and differentiate within the inner cytotrophoblastic layer into the vessels. Then later, these tertiary chorionic villi are very plump and diverse in their branching and structure. And again, we have the syncytotrophoblast layer or syncytotrophoblast layer in the outermost part, then the cytotrophoblast layer, then a thin layer of the mesoderm, and finally the fetal vessel. So this will change as we see the development occur. So gas exchange and nutrient and waste exchange can occur here, but not at the greatest efficiency. This will continue to develop so that it is optimized. So it is 2:50 at this time, so I'm going to stop at that point, and we'll begin there with the lecture on Friday and push on to complete this area of the lecture. I appreciate everybody's time and attention. Obviously, some individuals were not too pleased with their performance on the exam. Please communicate with us through the webmail so that Marco and I can get an assessment of what problems there might be and evaluate the potential negative impact of those numerous meiosis questions. I do apologize for their preponderance on the exam, and so that we can make an analysis. And still, very likely, we will offer one more opportunity at least for individuals to retake this exam, not in class, but on a computer, and the schedule will be open, and we'll provide at least one other opportunity, and we'll make those announcements on Friday. And then we'll determine how we will interface the score from that version or that attempt of the exam versus the one taken in class. So we are in the process of deliberating on that and making a decision. So please take some time to provide input now if you have a chance before you leave the classroom. Do it through the WebCT mailbox system, okay?